All right, the nervous system. Big system to know. You know, we switch around the order versus what the textbook is. The textbook has the nervous system after a couple like more units, like the muscles and stuff. The nervous system controls everything, so it makes more sense for us, and it makes more sense when we get into the muscle system. But you understand how the nervous system works and how everything works before we get into some of the other major systems in the body. It is the master controlling and communicating system of the body. It's what receives information, sends information, it controls how your body is running. So the cells need to send information and receive information that are part of the nervous system. Now, how we receive and send information is electricity and chemicals. So this is why they know if someone is brain dead. They have no electricity whatsoever happening in their brain anymore. The cells communicate with each other using chemical signals and using different or electrical signals and chemicals, which helps leads to some of the electricity. Now, when they communicate, it is rapid and it is specific. When you imagine, like if something, if your pencil is rolling off your desk, you can see it at the corner of your eye and reach for it before it even touches the ground. That's very, very fast, and like that you can grab on to something. And it's very specific, that your eyes are telling your brain, I need to move all the muscles in my arm and my hand and my fingers, and you can grab onto that, and it happens less than a second, like millisecond. And you usually have almost an immediate response to your body to anything. Again, we've got all these senses telling our brain different information, and we've got some type of response from our body that reacts on that. Now, the basic function of our nervous system, it controls everything in our body. Kind of the three breakdowns of that. We have to receive information. That's our sensory input. So we have all these sensory receptors all over the body that are detecting any type of change. I mean, they're detecting, looking around at you, but they're detecting if your body is getting hotter, if it's colder. They're detecting things inside the body. They're detecting things on the surface of the body. It's all of these sensory receptors all over the body that are taking in that information. We have to send it somewhere. That's the integration part. So we sense something, and you can sense, look, there's a glass of water. I'm thirsty. What should I do about it? That information goes through the sensory input. We'll talk about those sensory neurons. Sends it to the brain. This is the integration part of the nervous system. The brain figures out, what should I do about the fact that there's a glass of water there and I'm thirsty? Well, the brain sends information then through the motor output. This is where we're actually going to do something about it. These are called known as effector organs. What effect am I going to have based on the fact that I saw some water in a glass? I'm now going to have an effect. And I'm like, and this is my motor output. What does motor mean? What do you think? We're going to have movement happening. And in this case, my motor output is skeletal muscles. I can take my arm muscles, I can take some finger muscles, I can grab onto the glass, and I can bring it to my mouth and I can drink the water. That's the motor output. So you have sensory input going in, brain is the integration part. Let's figure out what we're going to do about that sensory input, and then we're going to have our motor output. What are we actually going to do? What is the response of the body to that sensory information? Divisions of the nervous system. We have a central nervous system, we have a peripheral nervous system. The central nervous system, two main parts of it, brain, spinal cord. We find these in the dorsal body cavity. What does dorsal mean? This is the back part. So in the space, you've got a cranial cavity up in the brain, and you've got a little space going through all those vertebrae. That's where your spinal cord sits. This is the whole integration and control center. This is where all the higher thought processes happen. This is where sensory information goes in, gets processed, and motor information goes out. It all happens up there in that central nervous system, in the brain and the spinal cord. Which means the peripheral nervous system is everything that's not brain and not spinal cord. So this is all part of the nervous system outside of the central nervous system. This is most of your nerves. So the things that are coming off of the spinal cord, things that are coming off and surrounding around the brain, these are where we have all of our nerves. So we have spinal nerves, the ones that are going to or from the spinal cord. We have cranial nerves, the ones that are coming off the brain. And we'll look at a couple of the nerves on the brain today when we look at the brain in lab. Now, of the peripheral nervous system, just looking at the peripheral, so not central, the peripheral nervous system, we need to bring, we need to bring information to the brain, and then information from the brain out to whatever our effector organ is. So our two functional divisions is a sensory division, we sense information. And the motor division, what are we going to do about it? We can also call them afferent and efferent. Now, you'll see words afferent and efferent in other things. Afferent always means towards something. And information, things that are in the sensory division or the afferent division, we are sending information towards the brain and the spinal cord. Anything efferent means exiting or leaving, and so we've got information leaving the central nervous system to go out to wherever. So I don't care whether you call it a sensory or whether you call it an afferent, they mean the same thing. Motor versus efferent, they mean the same thing when we talk about the different divisions. Going to, leaving. Now, so the sensory division, we have to send this information to the brain, all these things that we sensed, and of that we've got two different types of fibers, or we have more divisions. When we get into this nervous system, it's like divisions among divisions among divisions. So in our sensory division, we have somatic sensory fibers and we have visceral sensory fibers. Again, they're sensory. They're sending information to the brain and spinal cord for processing. It just depends on where those sensory fibers are coming from. Somatic sensory fibers are ones that are sensing changes in the skin, the skeletal muscle, and the joints. Visceral are talking about the major organs inside the body. These are all your visceral organs. Your intestines, your liver, anything else that's kind of found in your whole abdominal pelvic cavity. It's receiving information and sensing, that sensory fibers from those. So we just kind of divide it, depends on what parts of the body we're receiving information from the sensory fibers. So that's our sensory division, two different divisions depending on what it's sensing. And then our motor or efferent division, what are we going to do about that information that we sensed? So we're going to send information from the central nervous to our effector organs. It could be muscles, it could be glands, and we divide our motor division, and that's where we get divisions and divisions, our motor division into two divisions. We have our somatic and autonomic. I'm going to draw some of this out just so you see divisions and divisions. I'll draw out other stuff today too. So of our nervous system, of the ones we've had so far, what are the two major divisions of our nervous system? We have our central nervous system and our peripheral nervous system. And we've been talking about peripheral nervous system so far. What are the two main divisions of our peripheral nervous system? Sensory or afferent, and motor or efferent. And of the sensory division, what are the two different fiber divisions? Yeah. And of the motor division, what are the two divisions? Yeah, the somatic, autonomic. And then we're going to talk about the somatic and autonomic. Yes, there are two different types of somatic. One has to come, or sending information from the sensory to the brain. The other somatic is doing something about it. So we're going to look at the somatic nervous system, again, in the motor. So the somatic nervous system, we have somatic motor nerve fibers that are sending information to whatever effector organ it is. Anytime we talk about somatic motor nerve fibers that are going to go do something, the somatic are going to control skeletal muscle. Can you control your skeletal muscle? So yes, this is a voluntary nervous system. The somatic you can control. We have conscious control over skeletal muscles. I can tell my skeletal muscles to pick up the glass of water and take a drink, or I cannot. You have conscious control over the somatic nervous system. The autonomic nervous system, do you have control over it? No. Auto means it happens by itself, or it happens on its own. You don't have control over that. These are your visceral motor nerve fibers, controlling all the other internal organs. It regulates smooth muscles, it regulates cardiac muscles, it regulates glands, it regulates all the organs inside the body. It is involuntary nervous system. You have no control over it. And now we have two more subdivisions. We have the sympathetic and the parasympathetic that are divided under the autonomic. They work in opposition to each other. What one does, the other does the opposite. should really only have one triggered at a time. If one speeds something up, the other one slows it down, and vice versa. Now, do you remember from fun days, when does the sympathetic nervous system kick in? This is your fight or flight. So under stress, I'm just going to write it down here just for myself to know. This is under stressful conditions. They both start with the letter S. Stress, sympathetic. And so when does parasympathetic take over? Yep, under restful situations. There's the rest. Now, in your book, this is on page 388. It's another, I did that. This is the exact same thing as that. Page 388 in your book, in case you want to look at it. Again, it's just looking at the different divisions of the nervous system. You have your central nervous system, you have your peripheral nervous system. Of the peripheral nervous system, you have sensory and you have motor. And we also have somatic and visceral nerve fibers. Or somatic and visceral nerve fibers. Of the motor division, you have somatic. You can control, that's your skeletal muscle. And the autonomic, you can't. Autonomic, you can't. And now the autonomic, you split it up into sympathetic division and parasympathetic division. During stress or during activity, when you're under rest, is the parasympathetic. Does this look familiar? Again, this is still my favorite one. Just to kind of look at what is all happening in the nervous system with this organization. Again, you have to have sensory information. That's my stimulus, my input of information. There's a bear there, which I just saw in the news. There was a bear in La Crosse. In someone's backyard. They took a picture of it. I didn't know the name of the street. It's in the Shelby division, so on the outskirts of La Crosse. But in La Crosse, there was a bear. I like bears. I was going to say, my husband's dad last year got a bear license. Didn't get the bear. So he's back on the list. I want to see a nice bearskin rug. Anyways, I'm like, saw a bear. Most people would run. Other people would shoot it if they've got a gun handy. That's your information. So you have to go through the peripheral nervous system, which part of it is sensory information. So we're sensing the fact that there's a bear there. We're going to send that information to the central nervous system. What are we going to do about it? Let's process this information. So sensory information goes to the brain for processing, goes out to the motor division. Like, we sensed it, goes up to the brain processing. Let's do something about it. And then of that motor division, you have your somatic nervous system and autonomic nervous system. This you can control. You can control your skeletal muscle and you can tell yourself to run, or you can say, I'm going to, curl it up in a ball and pretend I'm dead. You can choose that. The autonomic nervous system you have no control over. This is your cardiac muscle, your smooth muscle, your glands. Your heart's probably going to start racing. You might start sweating. That's all stuff that you have no control over as you're deciding to run from the bear. Now, of the autonomic nervous system, there are the two divisions, sympathetic, parasympathetic. Which one's this one? This is the sympathetic. You're probably under a lot of stress when you see the bear. That's going to cause different types of sympathetic reactions, like increasing heart rate, things like that. That's kind of the basic organization of the nervous system. Now, if we zoom in and let's actually look at the tissue inside the nervous system. One, it's highly cellular. We have lots and lots of cells. Very little extracellular space. These cells are just jam-packed, really tight. They can communicate with each other very, very quickly, very, very easily. Not a lot of space outside the cells. And there are two types of cells. We'll look at them, but there are two cell types. There are cells called Neuroglia cells, and then there are neurons, also just nicknamed as nerve cells. So there are two cell types, Neuroglia cells and neurons. What does glia mean? Glue. They call this like the nerve glue. These are the cells that help hold everything together, hence their name. They've all got different functions. We're going to look at the 6 different neuroglia cells. But these are the cells that kind of hold the nervous system together. So we call them neuroglia cells. Now, I've got purple and I've got black up here. Why do I have 2 different colors? Yeah, the first 4, these are cells that you only find in the central nervous system. 5 and 6, the black ones on there, these are the cells that you find in the peripheral nervous system. They look different. Some of them have very similar functions versus central versus peripheral, but you only find them in one. You only find satellite cells in peripheral nervous system. You only find astrocytes in central nervous system, although they have similar functions. So let's look at them. We're going to start right down the row, starting with our number 1, astrocytes. Based on its name, what do you think? The word means star cell. It's named because it has all these cool little spikes that come off of it. I'm like, so it looks like a star, apparently. So they call them astrocytes. Of the neuroglia cells in the central nervous system, these are the most abundant, the most versatile, if you look at all of its functions. These one little star-shaped cells, most versatile, and it's highly branched, hence its name. You find them not hanging off by themselves. You find them clinging on to neurons. You find them near synaptic endings, and we'll get into a little bit of that. And you find them near and surrounding capillaries, which is what this picture is showing. Some of their functions. 1, it helps support and brace neurons. You're going to find them clinging to neurons. So it helps support the neuron that's there. It helps brace the neuron and keeps it in place. Because it also anchors to capillaries, it also helps play a role in exchange of information, or not information, nutrients between the capillaries and the neurons. So this one astrocyte, you only see it anchored to a capillary here. On the other side, it could easily be anchored to a neuron to make sure that that neuron has oxygen and nutrients right next to the blood supply. So it kind of keeps the capillaries next to the neurons, make sure there's lots of nutrients right there next to it. So it can anchor and hold on to a neuron, and it can anchor and hold on to a capillary and keeps them close to each other. Young neurons, as they're forming and moving to where they need to go in the nervous system, the astrocytes with their little branches can help move neurons to where they need to go so that they end up in the right spot. They can help control the chemical environment around neurons. There are lots of different ions, which we'll start talking a little bit about today. What are ions? Yeah, something that has a positive or negative charge, either lost or gained an electron. It's going to help control some of the ions that you find surrounding these neurons. Sodium, potassium, calcium. It helps control the concentration of some of these ions around the neurons. It can help respond to nerve impulses and neurotransmitters. Because it's anchored onto some of these neurons, we're going to have chemicals leaving some of these neurons. You're going to have electricity leaving some of these neurons. It can respond to some of that information. And it can influence neuronal functioning. So it can influence how that neuron functions. Mostly because it can also respond to some of these neural impulses and some of the neurotransmitters. Neurotransmitters, we'll get more into it, but I'm like, these are those chemical signals. When neurons communicate, it's through electricity and chemicals. Neurotransmitters are the chemicals. And so since they can respond to nerve impulses, the electricity, and neurotransmitters, it helps the neuron function correctly. It can help send information along, similar to what a neuron does. So these astrocytes, this first cell, very diverse in what it can do. It does lots and lots of different things. Big old picture. This is your book picture of it. Again, you've got it anchored next to a capillary, and here's one of my neurons. Here's one of the axons coming off in the cell body of a neuron, and you've got one of these astrocytes right there in the middle, anchoring it all together. The next neuroglia cell that you find in the central nervous system is known as a microglial cell. It's like a mouthful. The microglial cells, based on the part that it has micro in it, it's small. It is a small oval cell that has thorny processes that hang out on the outside. Those little thorny processes can touch on to these neurons. It monitors the neurons. It can migrate toward injured neurons, and it can even transform itself that it then becomes a phagocyte. It can phagocytize microorganisms and any debris that's in the nervous system. That becomes very important. Our immune system has very limited access to our central nervous system. It's a good thing. We don't want our immune system constantly, every time we have a fever and a mic and we've got white blood cells all over, we don't want it getting into the central nervous system and attacking when it shouldn't. So the immune system has very limited function in the central nervous system. So if you get anything that, into the nervous system that we need to get rid of, these microglial cells are going to help clean up the central nervous system. Third type of cell that we find in the central nervous system, known as ependymal cells. Some are ciliated, so I've got two different ones, A and B. Some are ciliated. The main thing that ependymal cells do, I don't have it on here, they help make, but not just make the CSF. Any guess what CSF is? Oh, it says down here. Cerebrospinal fluid. They help make the cerebrospinal fluid, and if they have cilia on them, which can beat back and forth, the cilia help move and circulate that cerebrospinal fluid. So today in lab, when we get into different parts of the brain, you're going to see some of these, they're called ventricles, these open areas inside of the brain. That's where you find cerebrospinal fluid. They're going to be lined with these ependymal cells that are making that cerebrospinal fluid, and then they're going to have cilia that help beat that cerebrospinal fluid around to help circulate it throughout the whole central nervous system. So they line the various cavities of the brain, they line the spinal column because you find cerebrospinal fluid there, and they form a permeable membrane, so some things can get across between the fluid inside these cavities and any tissue that you find around the actual central nervous system cells. So there's cerebrospinal fluid, and then there's tissue around the cells. These ependymal cells allows that fluid to get across wherever it needs to go. Then we have oligodendrocytes. It's another one mouthful. These are branched cells. They wrap around central nervous system fibers, and their main job is to insulate the fibers by creating a myelin sheath. So all the blue up here, this is all my myelin. This is one cell body of an oligodendrocyte, and it's got different branches that come off of it. That's where it gets the branched cells. So this branch that comes off, it forms a myelin, this blue sheath around this nerve fiber. Another branch that comes off creates more myelin, a sheath, a covering around other nerve fibers. And here's another branch that comes off that's having more myelin going around nerve fibers. And so the myelin that we find in the central nervous system, made by those oligodendrocytes. Now, the last two neuroglia cells are the peripheral nervous system cells. We have satellite cells and we have Schwann cells. You remember Schwann cells? Do you remember what they do? They also make myelin. I'm skipping ahead. So the two cells, the satellite cells and the Schwann cells, functionally they are similar to two other cells in the central nervous system, but we find these cells, they look different, and we find these in the peripheral nervous system. The first is known as a satellite cell. Here's my picture of it. Here's my satellite cell. It's named because they think it looks like this little round thing that's kind of orbiting, like a satellite, orbits around the top, and so they call it a satellite cell. It's like hanging out on top. Its main job is to act very much like an astrocyte. What's an astrocyte do? Yeah, the little stars. They do all the different things. Yep, they anchor the neurons to capillaries. They help move neurons where they need to go. They help regulate the fluid that's surrounding some of the neurons. Like the astrocytes in the central nervous system, they do a ton of different things. So do the satellite cells. They look different. These satellite cells look nothing like a star. They don't have those branches all over it. They kind of sound, like they're both outer space type things, stars, satellites. And their functions is very similar. It's just they look different and they're found in different regions of the nervous system. The Schwann cell, just like the oligodendrocyte, so that would be like its counterpart, different parts of it. The Schwann cell, just like the oligodendrocyte, it's written on there, makes myelin. It looks different. It's not branched. Like that oligodendrocyte, it's got branches all over it. The Schwann cell does not branch. It's one cell. I don't think I have a good picture of it. Maybe this would be closest. Here are the Schwann cell. It's like one cell, just in one little tiny area. There's no branching coming off of it, and it forms a little myelin sheath in one particular area. Now, this myelin covering of the nerve fibers, whether it is central nervous system, whether it is peripheral nervous system, I'm like, we'll talk about some of the different functions, but since it covers up and it kind of helps insulate these nerve fibers, if your nerve fibers get damaged, it helps kind of, protect it, just like having a Band-Aid wrapped around, any type of damage. The Schwann cells, kind of the myelin around these peripheral nerve fibers is like the Band-Aid. Like, it helps it so that it heals underneath it. So yeah, there were two types of nerve fiber, or two types of cells we find in the central nervous system. The 6 Neuroglia cells, and what was the other type of cell? That's one of the Neuroglia cells. There are 2 types of cells in the nervous system. There's the 6 types of Neuroglia cells, and what was the other type of cell we find in the nervous system? We have the neurons. So as we get into the neurons, a neuron is just a neuron. There's not 6 types. It's just a neuron. Now, they are called nerve cells. They are not a nerve, but they're called nerve cells, if that confuses a lot of people. They are not a nerve. A nerve is a whole bunch of axons all bundled together. And I'm like, these are just called nerve cells. These are the structural units of the nervous system. The neuroglia cells kind of hold it all together. They really do lots of different support for these neurons. The neurons are really the thing that actually do what the nervous system should be doing. They are large cells. They are highly specialized cells, and their main job is to conduct electrical impulses. They live a long time. Most live 100 years or more. They're amitotic, which means what? They don't replace themselves. So why is that good that they last 100 years or more? Yeah, you don't get any more, so whatever you got, hopefully it'll last your lifetime. They're not replacing themselves. There are a few exceptions. There are a few cells that if they're damaged, like in your smell, that they'll undergo a little mitosis. Not like a huge thing, but in general, they're amitotic. Whatever nerve cells, whatever neurons you have, whatever you're born with, it's what you got. Don't damage them. They also have a high metabolic rate. They need a constant supply of oxygen and sugar. They cannot live more than a couple minutes without oxygen. This is why I'm like, people that have heart attacks and strokes, and I'm like, time is of the essence. If they're not getting oxygen and nutrients to those cells, your neurons start to die, and they're not going to replace themselves, so it's irreversible brain damage. Now, all neurons have one cell body, and then they have a whole bunch of different processes that come off of it, but they've got one cell body. The cell body, I might get creative and draw today. I'm going to have to draw out other stuff anyways. The cell body, because in lab last time we previewed a neuron. There's my neuron, or my cell body. I'm not going to draw everything. It's the biosynthetic center of the neuron. Sounds cool. What does biosynthetic mean? Making living things. Biology is the study of life. It's making living things. It synthesizes proteins. This is where the synthetic part of it. It makes proteins, it makes different parts of the membranes, and it makes other chemicals. It's making things inside the cell. It has rough ER inside of here. I'm not going to try to draw that. Well, maybe I will. It has rough ER. What do rough ER do? They make proteins, and they ship them out somewhere. That's part of it. It's synthesizing and making proteins. Part of it helps make membranes, and some of that is the proteins that comes from it. They have a nucleus inside of it. It's got DNA in there. It also has, I got lots of colors today, a nucleolus. What does the nucleolus inside of it do? Yeah, it makes mRNA and anything. It helps ribosomes as well. Ribosomes. Makes the ribosomal RNA, so it helps make ribosomes, which will make more protein. It has a plasma membrane on the outside, phospholipid bilayer, just like other cells. This is a cell. It has a plasma membrane. This is the receptive region. We're going to have information getting into that cell body through the plasma membrane. We'll talk about some things that helps do it. Now, there are different places in our nervous system where a whole bunch of these cell bodies all clump together. Not talking about the processes that come off of it, but all of a sudden you find a whole bunch of cell bodies. If you are in the central nervous system, brain or spinal cord, and you have a whole bunch of cell bodies together, we call it nuclei. Isn't that great? If we have a whole bunch of these cell bodies all together and we're in the peripheral nervous system, we call it a ganglia. In lab, you'll see where a ganglia actually looks, it sticks out a little bit because you've got a whole bunch of these cell bodies. Now, of the processes that stick out of the cell body, we've got two main types. One, dendrites. The second, axons. I should really bring anything, Mike. The dendrites are short, spiky things that come off of the cell body, and you find lots of dendrites. Axons, so all of these little spiky things, these are my dendrites. You find one axon that comes off of a cell body, and it's longer than the dendrites. One axon. I'm really the same width. I just can't draw. Now, of the dendrites, this is where information is going in. So in dendrites, we find these a lot in our motor neurons. They're receiving information and sending it along to wherever it needs to go. You can hang hundreds, I drew like 10 up there, hundreds of short, tapering, diffusely branched processes on the neuron. So you find tons and tons of these little branches all over. This was a kind of a better picture. There's not even 100 on there. You can find hundreds of more of these little branches. They are the receiving region of the neuron. They are very much like the antenna on the old TVs. They receive information and they send it into the cell body. Just like the sensory information goes to the brain for processing and then goes out. The dendrites receive information, send it to the cell body, and then it leaves. Now, on some of the dendrites, which is shown in here, you actually even have little spikes that stick up. These are called dendritic spines. Any guess what they're for? Sometimes you just find little spines sticking up. You may have said it. Oh. Starts with an S. Surface area. It is surface area. That's what I'm like. I thought I heard it. I'm like, more surface area. On antenna. How many of you guys grew up with antennas on your TVs? Okay, at some point I know this is going to change. I can't use my analogy. You're all going to have, cable boxes. But as you grew up, and I'm like, if your antenna wasn't working very good, what'd you add to it? You added tinfoil to it. Why? Yeah. Why? Why'd you add tinfoil to it? Yeah, and I'm like, more surface area. The more tinfoil you added and did it in different directions and sometimes cut it and branched it and everything else, more surface area for receiving information, you get a better signal. Same thing. The more dendrites, the more dendritic spines, the more surface area, the more area you can receive information. So that's why you have hundreds of these things, and some even have little spikes. More surface area for receiving information. Now, the axon, so that's the main job of the dendrites. Receive information, send them to the cell body, and then that information will leave through the axon. So you have one axon per neuron. I need more hands. I'm like, it comes out. There's a little cone-shaped region. I tried to redo it. A little cone-shaped region that comes out. We call this the axon hillock. It's just a little part where it comes out, a little cone-shaped region on the cell body where the axon's going to come out. On some neurons, you have a very short axon. Some don't even have an axon. It kind of depends where in the body and what's it doing. Most are going to have one long axon that comes out. So in others, most are the entire length of a cell. Some neurons can be a meter long or longer. One neuron can be a meter long or longer. How many feet is in a meter about? About 3. Again, we're going metric to non. But there's about 3 feet. So imagine that's a really long cell. really long neuron cell, about 3 feet long. That means that an axon has to run that entire length. Where you're going to have the biggest neurons, which means they're going to be the ones that are found in the biggest nerves in the body, is where you go right from spinal cord right to an effector organ. You've got some nerves that are coming from your lower spinal cord that are going all the way down to your toes to control your toes and receive information and send information. That means that axon has to go that entire length too. So these long axons, if it's long enough, sometimes we just even call them nerve fibers. It's like just the longest axon, let's just give it its own name, a long nerve fiber. Occasionally, these axons branch. If I get enough colors. Sometimes they branch. We call these axon collaterals. And sometimes they branch. If I spell it right. Yeah, there we go. But at the end, whether it's the end of an axon collateral, or if it's just the end of the axon and it didn't have any other branching before that, we have lots and lots of branches at the end. We call these terminal branches. What does terminal mean? The end. So we call these terminal branches. Sometimes, you know, the very, very tips of these terminal branches, we call them axon terminals, the very end of the axon. And it also gets the French word of a terminal bouton. It's the French way of like a terminal bouton. It looks like a button, hence the name. That's just the end of these axons, at the very, very end. We get more into these when we get into muscles and what's happening at right here. But the very, very end. And they can branch up to 10,000 times. One axon can branch and branch, and you can have up to 10,000 different terminal branches at the end of an axon. It's a lot of branching. It can control lots of different things. Now, functions of the axons. We know information has to travel through it. So it's the conducting region of the nerve, the neuron. So information has to be able to travel through it. And when I talk about conducting, I'm generally talking about electricity. So it's going to help generate nerve impulses. It's going to help generate nerve electricity, which we'll talk a whole bunch more about next time. But it's going to generate nerve impulses. It's going to generate electricity along this axon. It transmits that electricity, that little nerve impulse, along the axolemma. If you remember, way back when, I don't remember if we talked about it yet. The word lemma, or the suffix lemma, just means a membrane. And so an axolemma just means the cell membrane found along the axon, or along the neuron itself. And eventually it goes all the way down to the axon terminals. There is a secretory region, which means secretion is going to happen at the very end of these. We have to be able to send information to something. It doesn't end and there's nothing here. There could be another neuron. There could be a muscle. There could be some other effector organ. And so we have to secrete something from these axon terminals. So there is a secretory region. And the main thing it's going to secrete is a chemical called neurotransmitters. And they all have different names. But it will secrete neurotransmitters. That's the chemical information that it's going to send to whatever's next over here. That neurotransmitter, again, the name it means nerve, and transmit is going to send information, is either going to excite another neuron to do something, or it's going to inhibit another neuron to stop doing something. Because these axons can branch so many different times, one neuron with all its little branching can have multiple conversations with different neurons at the exact same time. This allows us to, as you're writing down notes and listening to me, you're thinking, what am I doing later today? I got to do this. And you're thinking about other things, and you're breathing, and you're, smelling. And I mean, you've got lots of information traveling in and around your body and around your nervous system all the time. These neurons can handle multiple conversations with different neurons at the exact same time, because information is traveling to different areas at the same time. Now, the actual axon itself, unlike the cell body, lacks endoplasmic reticulum. It's not making anything down here, just sending information. It doesn't have a Golgi apparatus anymore. So any proteins it might need, especially ones found in the membrane, are all going to have to come from the cell body. And it makes them efficient transporters. It doesn't have all these extra organelles getting in the way. It's like, we've got our one job. Anything else that we don't need, let's get rid of it. I'm like, it's just in the way, don't need to be making anything, we don't need to spend the energy on making anything. They're just really efficient transport mechanisms. They just transport. However, if they get damaged, they decay. They can't make anything back on their own again. It's like if you kill that neuron, you've damaged even the axon part of it, it's going to die. Now, some of these axons have myelin sheaths on them, some do not. Myelin is a whitish protein lipoid substance. So it's kind of like a lipid and it's got proteins in it. Most of the time they're segmented, so it's not just one big myelin sheath. You generally find them in little segments, partly because of the Schwann cells are in segments. So you find myelin in them. I forgot my little clicker. If an axon has myelin around it, we call it a myelinated fiber. They don't all have myelin around them. So some do. Now, why we have myelin, whether it's central nervous system, whether it's peripheral nervous system, it protects the axon. I mean, this can be meters long, or a meter long, several feet long. It helps protect that axon, the skinny little thing that comes off that nerve, that neuron. It helps protect it and electrically insulates it. We're going to get a bunch into electricity on Thursday. So it protects it and electrically transmits it. Again, this one axon can be 3 feet long as it goes from your lower lumbar region all the way down to your toes. We want that electricity to travel along that entire axon. And it can help speed up how fast that information travels along the axon, which we'll talk more about how that happens later. So a non-myelinated fiber, it sends information more slowly. Like it doesn't have that nice insulated electrical insulation. That's why even, when you have any type of electricity in wires, you generally have an insulator around it, so you don't have electricity leaking out, because it can. Now, if you're in the peripheral nervous system, that myelin is made by Schwann cells. If you're in the central nervous system, what makes it? Yeah, the oligodendrocytes. So we're just going to mostly talk about the peripheral nervous system ones. Talking about the Schwann cells. Now they wrap myelin around the axon, and they call it in a jelly roll fashion. What of you know what a jelly roll is? It's, you lay some breading dough down, and you put some jelly on it, and you roll it up, and you slice it, and it gives that cool swirl. Same thing. It's also the same thing if you have a finger, and you take some gauze or something, and you just wrap it around it. You just jelly rolled it. So I'm like, we've got our axon, and we've got our Schwann cell, and it wraps around it in a jelly roll fashion. It's this constant wrapping around and around and around and around and around. Makes a really good insulator this way. I guess I don't really need to go back, but I'm like, so that myelin sheath, it's concentric. It goes around in circular layers of Schwann cell plasma membrane. So all of this myelin that goes around the axon. These Schwann cells, one cell forms one segment of myelin. So oligodendrocytes, they were a lot longer, they covered a lot more. One Schwann cell, got a nucleus and everything. One Schwann cell makes one, wrapped around jelly roll right there. It's horrible drawing. One little segment of myelinated sheath around that one little part of the axon. This is an actual picture, zoomed in 24,000 times. Here's an axon, so I sliced right through my axon, and I can actually see these myelin sheaths wrapping around that axon. Not really, but enough. Now, the myelination, the myelin around here, the plasma membranes, if you're in the peripheral nervous system, the plasma membranes have less proteins. Now, if you remember let me draw more, it's because I feel like it today. But if you remember in your phospholipid bilayer, draw a couple more, we had proteins shoved in there. What did some of those proteins do? A lot of them were channels or carrier proteins, and things could sneak through or move through. We don't want things sneaking through or leaving anywhere in these peripheral nervous system axons. So there are less proteins here. It makes them, so that we don't have any channel proteins or carriers Here, it makes them a good electrical insulators. Things cannot sneak out. We can't have ions. We'll really talk about sodium, potassium ions sneaking out where they shouldn't be sneaking out. So it makes them a really good insulator. However, if you notice when I drew my little Schwann cells that make myelin in these areas, there are spaces where there is no myelin. These are called your node of Ranvier. Each space is known as a node of Ranvier. It's a gap in your myelin. This is where an axon collateral might arise, is in one of those nodes of Ranvier. If there are any, it's where it's going to come. And most people think that's going to slow down the electricity if you've got gaps in myelin. It doesn't. It speeds it up. We're going to talk about why. But it speeds up electrical in-cell pulses by having those little gaps right there. It speeds along as you stand on one of the little moving. Kind of. But it speeds information up in those little tiny gaps. We'll talk about it later. That's what there's nodes of Ranvier. You just don't have any myelin in a couple little spaces done here. And there are some axons, even in the peripheral nervous system, that are non-myelinated. There's Schwann cells there. The Schwann cells just aren't making any myelin. They're not wrapping it around in a jelly roll fashion. So you might have some Schwann cells. There's no myelin getting wrapped around, and one Schwann cell might actually surround 15 different axons or different fibers. So all of a sudden you might have a bunch of axons all grouped together and one Schwann cell wrapping around all of them. It's just not making myelin. There's different places in the body where we just don't need quick, fast response to anything. Part of that is our intestines. There's really nothing life or death on why you need to digest faster, Mike. So information doesn't have to go to and from our digestive system very quickly. Now, in the central nervous system, the cells that make myelin are those oligodendrocytes. And those branches come off, and you can see them here. It's branching and covering 3 different axons in myelin. However, in the central nervous system, it can branch and surround up to 60 different axons. So there can be lots of branchings off this one cell body of an oligodendrocytes. There are still nodes of Ranvier. There are still some gaps. I don't show it on this picture. There are still some gaps where there isn't any myelin. Some of the thinnest axons, some of the thinnest fibers found in our central nervous system don't have myelin, so there's some unmyelinated axons as well. Usually they have other extensions of other neuroglia that are going to cover them, such as in the astrocytes that help protect it. The places where we have lots of myelin in our central nervous system, we call it white matter. Myelin is a whitish material. So where we have lots of myelin, we can actually see in the central nervous system, even in the brain, a whitish color to it. We call that white matter. We know there's lots of myelin found on the axons in those particular regions. The parts of the brain where we don't have a lot of myelin, so we have unmyelinated fibers, we call it gray matter, since myelin is a white. Like you might be able to see it today when we cut it through our brain and our spinal cord. Otherwise, we've got somewhere you can kind of see the different colorations of gray matter and white matter. Myelin, unmyelinated. That's a bigger picture. Now, of all of the neurons, we group them into 3 classifications, kind of just based on where we find them, what they're doing. So these neurons, their main job, they receive some information in the dendrites and they send it somewhere. If they are sending it towards the central nervous system, we call them sensory neurons. So this kind of makes sense here. If we are sending information from the central nervous system out to some type of effector organ, we call them motor neurons. And if they are found in the central nervous system, kind of receiving and relaying stuff in around that central nervous system, we call them interneurons. They're like the in-between. These are the ones hanging out, figuring out what we should do about the fact that we see that glass of water or whatever it is. 99% of all the body neurons are found in the central nervous system and are these, interneurons, the processing neurons. So we kind of just classify them on where they are, where they're sending information. Kind of makes sense. Then we classify them based on their structure. I'm not going to read through this. I'm like, just as you know, a multipolar neuron has multiple extensions coming off of it, so it's receiving more information and sending information out to multiple different areas of the body. This is showing, I'm like, one, it's receiving information, it's controlling different parts of one muscle. There's lots of different extensions. A bipolar neuron means it has two main extensions, one on either side. Bi means two. Again, it's a lot of receiving and sending information back and forth in a really short area. And something known as unipolar, it generally is just controlling one tiny little thing. This is kind of showing it's branching 3 times. A lot of times they just branch once, hence its name. And it's controlling one thing and one thing only. This looks like it's controlling like one gland found in the skin. So we kind of just look at their structures and how much they branch, which really is going to say what they all do. All right, some review. Pull out your color note cards. You're still going to draw some stuff, though. All right, the nervous system has 3 overlapping functions. Which of the following represents a logical sequence of these 3 functions? Is the yellow. You have to sense information. The integration is let's do something about it, and then the motor, or what should we do about it, and then motor is let's do something about whatever you sensed. While studying for an exam, you reach for a beverage. To extend your arm, your what nervous system is activated? It's the somatic, the skeletal muscle. You can control that you're going to grab, that cup or beverage or whatever. You're not stressed at this point. You're just reaching for a beverage. The door slams shut loudly and you flinch. After a few seconds, you realize your heart is beating very rapidly and forcefully. This response is the result of what nervous system? It is the autonomic. Can you control your heart beating? No, it's going to do it automatically. Complete the following analogy. Electrical wire is to electrical insulating tape, as peripheral neurons are to what? Peripheral neurons. There's the Schwann cells. The oligodendrocytes are the central nervous system neurons. Each neuron in our body has a lifespan of about how long? We all hope. It hopefully lives an average human lifespan, hopefully around 100 years, and hopefully you'll live that long. The portion of the axon that communicates with its target cell, whatever that might be, is the what? What part of the axon communicates with the target cell? Part of the axon, so it's a part of an axon, so just the part of the axon that communicates with its target cell is the axon terminal. At the very, very end, you've got these little axon terminals, and it's going to communicate with whatever is over here. Again, another neuron or a muscle cell or a gland or whatever it is. Oops. Oh, I had one more. The basis for differentiation between gray matter and white matter in the central nervous system is the presence of having what in white matter? You have those myelinated fibers. Myelin is white. If you have myelinated fibers, it looks like it's white matter. That's where we're going to end right now.